So we've discussed this whole concept of RNA making protein, and RNA does that using three forms, a T, an M, and an R. And this video, the last 3 minutes, finally is going to concern the translation of RNA to a protein. So let's hit the play button and pick up with the remaining portion of what goes on inside of your cells.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein.
So I'm going to pause it here because this is your mRNA, and your mRNA is the strand that you're seeing to the left and to the right. This is the ribosome, which is the factory that all of this takes part in. And then you're seeing the tRNAs that are floating around like little bees or little butterflies. So these tRNAs are carrying with them amino acids, and they're going inside of this factory, if you pay attention, and they are latching down the amino acids so the protein production can start to go on. You'll probably see the protein begin to get formed out of the ribosome here in a few seconds, but that's kind of the order, and those are the pieces and the parts and the players that's happening in this portion of the video.
Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So this is the protein that's being made. As she said in the video, this is going to be hemoglobin protein. And you're noticing that the amino acid has a primary sequence, and that primary sequence, or that protein, has a primary sequence. And that primary sequence is a chain of amino acids that are hooked up end to end to end. Secondary, tertiary, and quaternary structure is beginning to fold this amino acid chain into an actual 3D shape. And this 3D shape is very specific for that particular protein. So now you're starting to see the protein actually assemble itself, and this is going to be your hemoglobin, and this is just going to be a continuation of what hemoglobin is going to be doing. Okay, so that's the video for the what we're calling molecular visualizations of DNA. So this is putting a real-time perspective or a real-life perspective about what goes on inside of these cells, and what their purpose are and what do they do. Um, so that's what we're taking a look at least. Um, and we'll stop the video here and we'll continue on with some other discussions about DNA and maybe some more laboratory technique in the upcoming video or two.